So, Mr. Cimerman, you are a team leader at the EMBL Imaging Center. Can you tell us something more about the everyday work that you and your colleagues do?
Yeah, I'm glad to explain this a bit more in detail. So I'm the head of a service team at the Imaging Center for light microscopy, and we handle a wide range of projects. And I think one part of me is to coordinate this, of my activities is to coordinate this, to evaluate together with my colleagues, with the application specialists, the feasibility of a project, and then also together with the service manager to make sure how that this gets properly reviewed and then also executed. So I'm involved more on a high-level overview, but in the day-to-day, I'm following the projects that are going on.
And could you tell us more about some of the projects that you are currently overseeing and sort of their application in the everyday world?
Yes, that they are then much more along the lines also of my background, which is much more, of course, on the side of biology, of imaging. So we cover a wide range of different imaging technologies. Therefore, also the projects are quite different. Some need more time, some need less time. We have people that are coming for fairly concise visits, maybe of a week, so and can do everything in that time. That it may also not need necessarily a lot of analysis. On the other hand, we also have projects that run over several weeks where the people are really staying. This is where it's very important to be able to accommodate them, to give them a place to sit, to follow, to work through the data already while they're acquiring it, to work with the analysts also then for the analysis. And this is another form of how we are working. So it varies because in that field the workflows are very different and we adapt to the specifically needed workflow.
And then, if I understand correctly, your goal is to develop advanced light microscopy technology and provide such services globally, right?
Yes.
And can you tell us more about the application of this advanced technology? So where can it be used and how important is it for? The goal is also for our audience to understand completely what you're doing. So if we can try to put it on a very basic level so that everyone can actually understand technology behind this.
So yes, I'll try to really keep it on a basic level, and I think anybody that works or is interested in biology realizes that biology also can be very nicely represented by an image, very often by an image taken with a microscope. I think the fact that an image contains so much information is something that everybody can appreciate. But of course, you can go so much further. And we work with a wide range of samples. So anything that I think has biological relevance has also need of imaging. This can start with a big organism, but you can go smaller and smaller, or the other way around. If you think that normally organisms develop from a very small thing into a big thing, you can start with a single cell, you can go beyond a single cell, you can look at single molecules and find a lot of interesting things there that you can work on. You can, of course, also follow this thing as it becomes more and more complex. You can go into systems that are emulating processes of animals, of plants. This could, for example, be what's now called organoids, so simplified forms of organisms. And you can go to the whole organism and for every single thing you have an imaging technology, especially in light microscopy, that is actually matched. And we are covering all of those. All of those give images that are beautiful and highly instructive. And highly instructive in the end means we need to come out with numbers. And that's, I think, the currently most interesting challenge, and also to introduce artificial intelligence into this deep learning and derive information out of something that has a beauty, but as I said, also a lot of information. Many of our systems are laser-based. They're also technically very, very interesting. And we start really with single molecules and we can go all the way to a full-fledged animal in motion.
Mm-hmm. That sounds really interesting. I also wanted to ask you if you have already started sharing your knowledge and expertise with other colleagues and on a global level. maybe to tell something more about the overall international cooperation that the EMBL has.
Yes, I think EMBL has overall the mission to share knowledge, to distribute knowledge, and this is also a strong focus of our activities. This can take multiple forms. Sometimes it is, especially for more established technologies, it is important to have training courses, something that you can already put in a curriculum and share. This is very important. This is something we are doing. This is something that's being done in different forms at EMBL for all technologies. In addition, we are more and more also offering job shadowing, where, let's say, this is more of a one-to-one version of this, where a specialist, somebody that's already quite knowledgeable, wants to understand a new technology or new workflow and can just follow the expert around. We're also
it's like a sort of mentorship, right? Or something similar?
Mentorship is maybe too integrated because it would later on maybe focus also how this is applied, but we are exchanging knowledge really on a one-to-one basis where, for example, a very good example of this is MinFlux technology, which is now a very recent form of light microscopy, more specifically of super resolution microscopy, and it requires an understanding of how to prepare the sample. It requires a lot of understanding how to optimize the measurement. It requires also a grasp of what data are coming out and how you analyze them. And there are now more and more institutes that are incorporating this into their technology portfolio, but they are also very often talking directly to us, and we are sharing our knowledge on a one-to-one basis with them. So either they come to us, this sometimes happens, and that's, I think, very productive. Sometimes even in this case, application specialist, he goes to the other installation. And so we very actively exchange this knowledge. And in this case, of course, it's in a very targeted, one-to-one, customized way. And all of that is really exciting and very useful.
And before coming here, I mean, to the EMBL, you also used to work in Barcelona. You were head of the Advanced Light Microscopy unit in Barcelona. So what was it that you did there, and how does it differ from the work that you do at the EMBL?
So my work in Barcelona was very nice. I enjoyed it a lot. It was, I think, also very useful, as core facilities are useful. So providing technology or technological expertise to people that have very interesting questions in biology, this I found always extremely satisfying. This is something I've been doing through my whole career, and I moved into this more independent position in Barcelona. It was more inward-looking, and I enjoyed this a lot, really working with researchers in there that are really still doing the experiments, so working with people at the postdoc level or actually PhD students, and sharing the excitement of this, and establishing new technologies. All of this is something I already did there. I really enjoyed this work. I think the reason why I changed to the imaging center later on was because the imaging center concept includes this very strong focus on external service provision, which is different because you have to take other aspects on board. You have to prepare differently. You have to provide a different kind of infrastructure. And then you have to make sure that everything works in the time that you plan it. So there are logistical challenges which I think need to be appreciated and which I wanted to see.
And in the light of the emerging technologies and also artificial intelligence and all the great and also not that great things that AI is bringing with itself, what do you think is the future of biology and the future of your work and maybe your own future in this field in some, I don't know, 10 or 15 years from now? How is it going to look like, in your opinion?
It's very hard to look that far forward. It's probably impossible. However, I can say something with conviction. There's no end of the road in sight. This is actually, I think, a blessing that there are so many possibilities still to go forward, and technologies at some point may stagnate on a certain aspect, but other things will open. And no matter what, there's so many questions in biology that you will never run out of things to apply it to. So I think everything that happened during my whole career, so as long as I'm really interested in biological sciences, has actually only been finding more doors to open, and nothing was ever closed, and we never left anything. Sequencing led to the whole genome project. The whole genome project basically just told us that genes are not everything. Then we now are working on genomic regulation. This will still be work of decades. The layers of complexity we were just opening doors, and technologies are following this, and like you cannot run out of things to do. And I think this is and this will be in 15 years. Where this is influenced by AI is beyond me to say. I was now deviating a bit, my apologies, but I think we will still improve imaging technologies. We will very strongly learn how to use different modalities more efficiently together, how to also use different data qualities, and that's maybe something where also AI or any kind of improved data handling would be important. You have, for example, data from mass spectrometry, which tells you a lot about a protein, about the composition of a protein. How does this relate to the image? This is something that right now is extremely difficult to transfer, and it is not a technical challenge. You can get a very good image, you can get a very good data set. These two data sets are related, and we are still in the process of relating them. And I think this is where, for example, bioinformatics has still a lot to do, and it will also become more and more multidimensional, going from, let's say, a linear sequence, which already is extremely challenging if you think about what happens in genomics, to multidimensional data, to connecting all of this. I mean, this will not end. In 15 years, we will just be at the beginning.
And that's the beauty of science, I guess.
That's the beauty of science.
And one last question. Is this your first time in Belgrade, and have you been here before? What is your, maybe, some impressions about the city and also about colleagues from Serbia?
Ovo je mi prvi put u Beogradu.
Bravo.
But I know a little bit let's say, Central Europe in different aspects. This is my first time in Belgrade, as I was saying. I enjoyed it very much. There are aspects I'm familiar with and that are still excitingly different. And the interactions I had up until now were just really exciting. I have a lot of respect for the qualifications and the skills that can be found here. And it's really always exciting to get into contact. Some people I know, I've seen also some in science, you always find people that you met before. And realizing that there's so many new people to meet, this is exciting. And I really liked Belgrade. I had only a very short walk around, but I can put it a little bit into context, and I actually want to see more.
Okay, please come again then. We will be your hosts.
Thank you so much.
And thank you for the time, and thank you for the interview. It's much appreciated.
My pleasure.